Welcome back for more AP Biology. Today we're going to be dealing with how we control and regulate all of those genes that we dealt with last time, especially with biotechnology. So, like I said, what we're going to look for is how do we control genes, but in particular with protein structure. And this is something that the College Board likes to test on. And there it is from your book. So, it helps if we put some things into context. So if I look at bacteria, the simplest thing that we can look at, what do we see when we look inside of their cells? Well, we see that they have cytoplasm. We'll see that they have ribosomes. We'll see that they'll have junk in there. They'll have all of their DNA in one primary spot that we'll call the bacterial chromosome. It's circular. And there may or may not be plasmids present. So plasmids are those small circular pieces of DNA that we dealt with last time. So that's all we happen to notice. There's no buffer. It's a cell by itself, which means they need to be able to control their genetics really fast. So when we look at the structure of their genes, they turn out to fall under that category. The entire structure is referred to as an operon. And operons turn out to have some parts. One of those parts we've already seen, and it's referred to as a promoter, or the starting point for transcription, or the TATA box. So the promoter turns out to be where RNA polymerase will bind, but we also have a control section referred to as the operator. The operator, like if you operate machinery, you determine what it's going to do, meaning does the machine work or does it not? The job of the operator in terms of an operon is it is the thing that will determine does RNA polymerase get to bind or does it not get to bind. So why would you do that? Well, I don't want to have all genes working at all the time. So what I can do is, if I have some type of regulatory element, so some protein that blocks the operator, the result is the promoter won't, it becomes non-functional because RNA polymerase can bind there, but it's not going to be able to move past the operator. But if I could steal away the operator, now it will work. Or maybe I could have something that binds to the operator that will make it easier for RNA polymerase to bind, and then the result is I express the genes, or I don't, because the thing didn't bind there. We have two different ways that we can make this thing work. I could use the operator to block, but I could also use the operator to help. Well, how would I undo those two pieces? All I have to do is modify the regulatory protein, and then it works, or it doesn't work. There are lots of examples of this. So there are things called the lac operon, the R operon, and the trip operon that we can go into, but the details really aren't that important. If I can make the operator, put something onto the operator that will stop RNA from polymerase from binding, the result is no gene expression. If I can put something on the operator that will help RNA polymerase bind, we'll get gene expression. If that thing that blocks is removed, gene expression, but if I remove the helper, no gene expression. So I have two different ways I could view this. I can use the operator as a blockade, or I can use it as a helper. Well, what else can we do? Something that bypasses all of this would be the use of plasmids, especially in biotechnology. So when we're doing DNA manipulation, what we can actually do is take those plasmids and actually put viral promoters onto them. Why would I do that? Viral promoters turn out to be better at helping out RNA polymerase to bind than our bacterial or eukaryotic promoters. The result is if I really want a gene to get expressed, I use viral promoters and I can put those onto plasmids. But what about us? Because we're not bacteria. Well, if we look at most of our DNA, what we'll notice is some of it is used to make proteins. It's that purple wedge. We have that blue wedge, which doesn't code for protein. We have that red section, which is just a whole bunch of random stuff, randomness and repeats. And then we have a little wedge in there that are for things that look like they're genes, but turns out they're not functional. A lot of the non-coding DNA turns out to make things like, well, parts of the non-coding and part of the protein coding turn out to make just RNA. But a vast majority of our genetics isn't really used to make protein. So if I look at what we know, we get this list. And it has commas in there because it's European, and in Europe you don't use periods, you use commas as points. So when it says like up here where it says extracellular matrix proteins and it says 0,4, that's 0.4% to us. So we can see where most of our genes turn out to be thrown in, but as of the time that this picture was taken, which is a few years ago, a quarter of our genes, we had no clue what they did. So we were working on that, and that's one of those things that the gene chips could do. So how do we control our genetics? Well, we don't need a one-stop shop. Bacteria, they need a one-stop shop because they need to be able to change on the dime. We don't need to do that. So I can regulate our chromatin and make it easier or harder for transcription to occur. I can mess with transcription. I can mess with translation. I can then mess with the protein after the fact. We actually have multiple places where we can control what's happening. Let's do the scariest one first. It's referred to as epigenetics, which is the role of the environment. So when we look at the environment, for reasons that we're starting to pretend to understand, I think, would probably be the safest thing to say, we know that what we can do is we can take our chromatin, which is protein and DNA. In particular, the name of the protein is called a histone. There's 7 or 8 of them. That you have this ball of histones and your DNA coils around it and makes this little structure called a nucleosome. And all of that we call chromatin. So if I look at chromatin, either looking at the proteins that hold it all together or I look at the histones themselves, what I can do is I can either loosen it all up, which makes it easier to turn genes on, or I can tighten it up, making it harder to turn genes on. One of the ways that we can do that is we can add acetyl groups, so acetylation, which is under where it says genes on. I can remove things called methyl groups, which is demethylation. Those two things, for whatever reasons, tend to make it easier for transcription. If I want to make it harder, what I would do is just do the opposite. I remove the acetyl groups, so deacetylation, or I add on methyl groups or methylation. What causes this? The environment does. And it turns out that these methylations and acetylations, or deacetylations and demethylations, can be inherited. The result is Jean-Baptiste de Lamarck from way earlier in the year was right. You inherit what you get during your lifetime. You inherit environmental factors, which is terrifying. And we're just starting to understand this. So what do we have? Well, we also have a promoter. We don't have an operator, but we do have a promoter. And we actually turn out to have a variety of regulatory proteins and regulatory elements. Some of those elements are going to help RNA polymerase bind, so we call them enhancers. Some of them are going to stop it. We call them silencers. All of those things that are going to either help or hinder are given a generic name of a transcription factor, or a TF, because they are things, they are factors that will determine do we have transcription or not. And we don't need to necessarily have all sorts of details, but the big point is we happen to have things that can either help or hinder RNA polymerase from binding. We can accept that it's never just one or the other. We have complexes of these things. The other bit, which I didn't point out, so I'll point it out now, is we only deal with one gene at a time, with us, eukaryotic organisms. Bacteria, if you saw on the operon, if you noticed that it had like an X, Y, and a Z, they deal with clusters of genes all at once, because if the environment changes, they need to have a whole bunch of genes to deal with it. Well, smart money says just stick all the things that you need together, all together. That's what bacteria turn out to do. We turn out not to do that. Well, why would any of this matter? Why do we care about how we turn genes on and off? Well, it turns out the ability to turn genes on or off can lead to cells becoming different. Because if I can turn different genes on or off, the result is I can make different types of cells. And that refers or gives a determinate fate to cell types. Well, what's the cell that starts this all off? We call them stem cells. because that is the stem, the starting point for all of the diversity of the cells. How would that work? So you have adult stem cells. So we have the cell by itself. You get 2 of them, hooray, they're practically the same. But if I were to make a double-layered ball, the result is the outside has different experiences than the inside. They're exposed to different chemicals. Chemicals, the environment, influences that phenomenon of epigenetics, manipulating the chromatin. That will result in different types of cells expressing genes differently, which will result in different type of chemicals being secreted, transcription factors, that will then turn on and off other cells, and as this multiplies, we get a cascade effect, ultimately leading to various types of cells. Because they are in different environments mean different signals mean different genes are turned on or turned off, which means ultimately they're set on a path. And if cell 1 gets a different signal than cell 2, or if this cell turns on a gene and this cell doesn't, this one ultimately will behave differently than this one will. And what we're learning to do with cell signaling is how can we move backwards through time? How can we figure out the order of what genes you turn on and turn off? Because if you can do that, we can reverse engineer and go back towards stem cells. And we're starting to learn how to do that. Well, all of this stuff, this turning genes on and off, well, that leads to cell diversity. So, why do we care about cell diversity? Why do we care about any of this gene regulation junk? Well, if I look at bacteria are single-celled for the most part. They can live in clusters, but they're single-celled. So when we look at them, the way that they increase their diversity actually tends to be rather limited. They could have a transformation, which is this middle section. That's the lab that you're doing, and that was the transformation principle that was shown by the Griffith and Avery experiment with the Streptococcus pneumonia bacterium. We could have viruses that actually spread genetic information. We refer to that as transduction, and it has to do with the way viruses replicate. It's usually an accident. But you could also have bacteria purposefully trading information, and that's a phenomenon referred to as conjugation, and that makes use of plasmids. And the name of the plasmid is actually kind of weird, so we're not going to go there. But bacteria have a few ways that they can increase their genetic diversity. The result is they're still the same type of cell, though. So when we look at them, the cell looks the same, just their internal components are different. If we look at us, we turn out to have a whole bunch of options. Because they obviously have, and I didn't show it in that last slide, they have mutations. We have mutations, but we can have DNA mutations and chromosomal mutations. We also happen to have sexual reproduction. So that results in, you have independent assortment of the chromosomes. We have crossing over events, shuffling the actual components of the chromosomes. We also have random fertilization. So we don't know what sperm and what egg are going to run into each other, which 2 gametes, the plus or the minus gamete, run into each other. Because some organisms don't have males and females. Or at least we don't use those words. We just say plus and minus. So why do we ever care about any of this? Well, it turns out, at least with us in eukaryotes, there's a very strange pattern that seems to exist of, if I look at a parasite, if you have a host and it's being plagued by a parasite, it can evolve to run away. And the result is, haha, I have escaped the parasite. And then the parasite will evolve to match up, because evolution is generation to generation. So we just have natural selection events. And the organisms, the parasites that can break into the host, are the ones that get to reproduce again. And those that don't die off. And the result is, you have the host, it happens to have parasites, and the ones that can break away from the parasite, they survive better, which now means the parasites, the ones that can break into the new version of the host, get to survive. And the result is you get this running away, just stay exactly where you are, which is referred to as the Red Queen, or the Red Queen hypothesis. Faster, faster.